The development of the CRISPR-Cas9 system, repurposing the prokaryotic adaptive immune system for genome engineering. The ability to eliminate, replace, or modify sequences within the genome in vivo in a highly targeted and reliable manner has a broad range of important applications. Double-stranded breaks, DSBs, are usually repaired by the endogenous cellular DNA repair pathways, NHEJ, non-homologous end joining, or HDR, homology-directed repair. Both of these repair pathways can be exploited to engineer the genome. A number of genome engineering techniques have been developed and used over the years. These all function with the same principle. A nuclease is guided to a specific sequence within the genome, inducing a DSB. These include zinc finger nucleases, ZFNs, a class of engineered DNA binding fusion proteins, talons, transcription activator like effector nucleases, another set of chimeric proteins with programmable DNA binding domains, and lastly, the CRISPR-Cas9 system, a genome editing method derived from the prokaryotic RNA-guided defense system. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR repeats were first discovered in the E. coli genome as an unusual repeat locus, the significance of which was appreciated later when it was realized that the spacer sequence within CRISPR loci were derived from viral genomic sequences, and the CRISPR-Cas system's role in prokaryotic adaptive immunity was realized. CRISPR loci are made up of short, partially palindromic DNA repeats occurring at regular intervals, direct repeats, which alternate with variable sequences known as spacers, which correspond to sequences within foreign genetic elements, protospacers, such as plasmids and viruses. Clustered CRISPR-associated Cas genes flank these loci. CRISPR-Cas systems and the elements are incredibly variable, differing broadly in occurrence, genes, sequences, number and size across the prokaryotic genomes. CRISPR loci find around 45% of known bacterial genomes and 83% of archaea. These sequences can evolve rapidly in response to local predators and therefore vary among isolates of related strains. The CRISPR-Cas immune system functions in three main steps. The first step is adaptation, where short sequences or protospacers from the invading viral genome are copied as spacers between repetitive sequences in the CRISPR locus of the host genome. This is followed by CRISPR RNA, cRNA biogenesis, where CRISPR arrays are transcribed and processed by Cas endoribonucleases within repeat sequences to generate small interfering cRNAs. The last step is targeting, where cRNAs guide the Cas nucleases for specific cleavage of homologous sequences. The match between the cRNA spacer and target sequences specify the nucleotidic cleavage of the invading nucleic acid. CRISPR-Cas systems have been classified into three major types, type 1, 2, and 3. In type 1 and 2 systems, nucleotides adjacent to the protospacer in the targeted genome are known as protospacer adjacent motifs, PAMs. This is essential for the Cas enzyme to cleave its target DNA, enabling the CRISPR-Cas system to differentiate between invading viral genomes and the CRISPR locus in the host genome, which does not incorporate the PAM. Type 2 CRISPR-Cas systems have been adapted as a genome engineering tool. The system requires three main components, the Cas9 nuclease, cRNA, and trans-acting CRISPR RNA, tracer RNA, which is critical for cRNA maturation and recruiting the Cas9 nuclease to DNA. This three-component system was further simplified by fusing the cRNA and tracer RNA together into a single guide RNA, sgRNA. Three main versions of the Cas9 nuclease have been adopted of genome editing and engineering. Wild-type Cas9 is an RNA-guided endonuclease that catalyzes blunt-ended dsDNA breaks. Cas9 cuts the two strands of a DNA target with two independent nuclease active sites, RIVC and HNH nuclease domains, to cut each DNA strand. Mutant nickase variants of Cas9 such as Cas9D10A and Cas9H840A, with mutations in RIVC and HNH domains respectively, cleave only one DNA strand. These reduce the potential for off-target cleavage of DNA and favor more predictable changes made via the result of HDR DNA repair, thus reliably introducing mutations into genomes. The last type is nuclease deficient dCas9, which has both RIVC and HNH domains mutated and can sequence specifically target any region without cleaving the DNA. This can be fused with various effector domains, for example transcriptional activators, repressors, and fluorescent proteins, for use as a gene silencer, activation tool, or as a visualization tool. RNA-directed Cas9 genome editing enzymes perform with comparable or greater efficiency relative to ZFNs or TALENs, and offer several advantages over these protein-guided systems. Targeting new DNA sites with Cas9 only requires designing an sgRNA complementary to the target DNA, which is much faster, cheaper, and easier to implement compared to developing new ZFN and TALEN proteins for each target site. The Cas9 platform also allows simultaneous editing of more than one site. This is known as multiplexing, and done by the expression of more than one guide cRNA or sgRNA.